Okay, so I might get started then. Thanks, Renee, for the introduction. So I'm going to present some of the work I've been doing here at the University of Queensland on RNA interference-mediated control of Myrtle Rust. And so this is part of my Advanced Queensland Research Fellowship, which is partly funded by the Queensland Government. So what we're trying to do, it might just be a bit slow here. Yep, here we go. So we're trying to look at environmentally friendly control methods for plant diseases. That's because traditionally plant diseases have been controlled predominantly through the use of pesticides or genetic modification, both of which have a number of issues associated with them. So pesticides suffer from issues of the pest developing resistance. They can leave chemical residues on our produce. Chemicals can run off into the environment, so into our waterways. And these pesticides can lack specificity, so they can also target beneficial organisms. The other problem is that new chemicals are just not forthcoming. The development of a new chemical actually takes about 10 years at a cost of $250 million. The genetic modification approach, or GM, avoids the use of the chemicals. However, there are issues of community acceptance. They can have some environmental impacts, for example, development of weed species. There are regulatory hurdles to overcome. And not all crops can be transformed, so this option isn't available for every plant. And this approach also costs a lot of money and it takes a very long time. So to develop a pest or pathogen resistant GM crop, it takes about 13 years of research and development at a cost of $136 million. And these approaches aren't really applicable to controlling diseases such as Myrtle Rust in natural environments. So we've been working on a new technology, which is known as RNA interference vaccines, or RNAi vaccines, also known as RNA biopesticides, to control pests and pathogens. And so this was first developed to target plant viruses, hence the name. And so I've just shown an example here of this approach being used to target a virus. But it now can be used to target other pests and pathogens. So it's a bit similar to some of the GM approaches in that we actually apply pest or pathogen-specific double-stranded RNA to the plants. But in this approach, we're just spraying this RNA onto the plant. Usually we spray it, you can apply it through other ways. Whereas in the GM approach, we're actually integrating bits of DNA into the plant's genome, which will then go to produce this double-stranded RNA in the plant. But anyway, what we do is we spray this RNA that we've synthesized against this pest or pathogen onto the plant. And then when, in this case, a virus comes to infect the plant, this double-stranded RNA is recognized as being foreign, and it's actually degraded by the plant. So it's chopped up into these small interfering RNAs, which can then go and target any homologous RNAs, any RNA with the same sequence, in this case, the viral RNA, and that RNA will get degraded by enzymes present in the plant. And so this leads to protection against the virus. So when the RNA is applied, the plant is healthy. Without the RNA, in this case, these lesions are formed. And the beauty of this approach is that the specificity to your target is very high, because we can use bioinformatics to check the sequences that we're synthesizing to make sure that they're only going to be homologous to our pest or pathogen of interest, and they're not going to target other organisms in the environment. If there is an issue of the pest or pathogen developing resistance, we can very easily find a new target sequence and deploy that to spray. And as I mentioned before, it's just a finite amount of double-stranded RNA being applied, and there's no chemical residues left on the plants like the traditional pesticides. And it's not GM, which is a huge benefit. And so in Australia in 2019, Parliament actually modified the gene technology regulations just to clarify that topically applied double-stranded RNA is exempt from the GMO regulations. So basically just clarifying that as long as the RNA cannot be translated into a protein, it doesn't get integrated into the organism's genome and it doesn't give rise to any infectious agent, then this approach is not GM. So as I mentioned, this has now been shown to work against insects and fungal pathogens as well as viruses. So I've just got some examples here. So on the top left, this group was targeting the Colorado potato beetle. And so they found that when they applied the double-stranded RNA, which was targeting the actin of the beetle, the beetles were all quite stunted in growth. With GFP double-stranded RNA, there was no effect on the growth. So that shows that it was actually the sequence-specific double-stranded RNA that was having the effect. And so I've got some fungal examples here. So these leaves here are canola leaves. This group was targeting the fungal pathogen Sclerotinia. So they tried some different double-stranded RNAs targeting Sclerotinia and found that the lesion sizes were reduced compared to in the control. So they saw some protection. This group here was targeting Fusarium graminearum on barley leaves. So the bottom here is showing the lesions developing when there's no RNA being applied, just the inoculum. And in this top one, when the Fusarium-specific RNA was being applied onto the barley, the lesions did not develop. And the approach has also been shown to work for post-harvest diseases. So this group was targeting grey mold disease, Botrytis grey mold disease, and they were trying to protect fruit and some vegetables. So they sprayed these fruit with the double-stranded RNA targeting botrytis, and they saw a bit of a reduction in the disease compared to the fruit that hadn't been sprayed with the RNA. They also tested lettuce, onion, and rose petals. And this has also been shown to be active against some rusts. So it's been reported that RNAi vaccines are effective against switchgrass rust, shown here. So these were all the different constructs they tried. A couple of them looked quite promising at reducing the disease on the rust, on the grass. And this group here, we're targeting Asian soybean rust. So this is with water, and these 3 leaves have been sprayed with the double-stranded RNA targeting soybean rust. And you can see there's a big difference between the control and the treated leaves. So this is all great in the lab, but what are the applications for the field? Double-stranded RNA is quite unstable, so it will degrade over time on the plant surface. And this is because there are nucleases present, so enzymes that will break down the RNA. Sunlight will break down RNA. If there's rain, the RNA can get washed off the leaves. And this gives it a very short protection window of only a few days. However, here at the University of Queensland, we've come up with a solution for this. So this was work performed in Nina Mitter's lab at Quaffie, together with researchers at the Australian Institute for Biotechnology and Nanotechnology. And so they actually made some clay nanosheets, which the RNA binds to the clay nanosheets, and it's actually protected from degradation. And so these results here are just showing the double-stranded RNA on the leaves. So with the naked RNA, when it was just applied on its own, the RNA was degrading after 5 days. So there's a very faint signal, but this is all starting to degrade. When the RNA was applied together with the nanoclay, and we actually call this bioclay. Bio is referring to the RNA component, and clay is referring to the clay nanosheets. The RNA could still be detected after 30 days. And after washing to stimulate rain, the RNA on its own completely washed off the leaves, whereas when it was in bioclay, it was still bound to the leaf. And a virus challenge was performed 20 days after applying the bioclay, and the leaves were protected against the virus. So we can basically harness this platform if we can find some double-stranded RNA sequences that we want to deploy against certain pests or pathogens. And this leads me to the work we've been doing on Myrtle Rust. And I have to acknowledge my very talented honours student, Rebecca Degnan here. Most of this work that I'm presenting is part of her honours research project, and I'm supervising Beck in collaboration with Louise Shuey at the Queensland Department for Agriculture and Fisheries, and Alistair McTaggart, who's also here at UQ. And this is Beck in the lab with some frangipani leaves that are covered in rust. She's very happy because she's found a lot of rust. So fungi are a bit different to viruses. Fungi are eukaryotes. And as I mentioned before, the RNA interference pathway is actually highly conserved across most eukaryotes, and it has been found in a lot of fungal pathogens. So to begin with, we just wanted to see whether the RNAi machinery was present in Austropuccinia psidii, which is the causal agent of Myrtle Rust. And so we did a bit of bioinformatics searching, helped by Alistair, and we pulled out a dicer gene, 2 Argonaut genes, and an RNA-dependent RNA polymerase, all of which encode proteins that are involved in this RNA interference pathway. So this led us to think that perhaps the fungi had an existing RNA interference pathway. And so we have 2 approaches here. Basically, when we spray the double-stranded RNA onto the Myrtaceae plant, the RNA could get taken up by the plants and processed by the plant's RNAi machinery. Or if the rust spores land on the leaves and happen to pick up the double-stranded RNA, perhaps they would process the double-stranded RNA themselves. So we wanted to set up some spore germination assays as a first step. So Beck just did some different germinations at different temperatures to find optimal conditions for spore germination. She did this all on glass slides set up in petri dishes to maintain the humidity. So she basically had agar drops on the slides, and then she dropped on a spore solution and then monitored germination after 24 hours. And just a bit frozen here. Here we go. So these are the results. So she found that the optimal germination temperature was 16 degrees, but they actually germinated as low as 8 degrees, which was quite surprising, we thought, and up to 25 degrees. But as you can see in the photos, there was really minimal germination at these higher temperatures. So that was an interesting conclusion in itself, because it made us think that maybe this pandemic strain might have a greater impact in cooler climates than was previously believed. But we were basically just interested in figuring out the optimal germination conditions for our in vitro assays. So the first test we did was to see whether the rust spores could actually take up double-stranded RNA. So we labeled the double-stranded RNA with some fluorescent nucleotides, so some Psi3 nucleotides. So the orange that you can see here is actually the double-stranded RNA fluorescing. So these images were taken with a confocal microscope, with a fluorescent confocal microscope. So the germinating uridinospores took up the RNA really well. These aciospores also took up the RNA when they germinated. We didn't see much uptake with the teleospores, but that was kind of expected because these are resting spores. They probably have a thicker cell wall and might not take up things so well from the environment. But we were quite happy that the uridinospores took up the double-stranded RNA because they're the spores that we usually want to work with. They're the ones we use to infect the plants in our assays. So Beck did a quick test to see whether rust-specific RNA causes any effects on spore germination. So we chose about 10 targets. So most of these were essential rust genes that we were targeting. We also had some genes that were only expressed in the haustorium. And so basically Beck mixed this Myrtle Rust specific double-stranded RNA with the uridinospores and set up the same germination assay on those agar drops. And then after 24 hours, just counted how many spores had germinated. So this is the water control here. It did look like there was a slight trend down with spore germination, but there was so much variability. It was really hard to draw any conclusions from this experiment. And there didn't seem to be much of a trend with the germ tube length. So we decided to move on to some detached leaf assays. And this actually worked much better. So this was all done with Syzygium jambos leaves because this plant is very susceptible to myrtle rust and develops these nice pustules, so it's very easy for disease scoring. So these leaves here are the beta tubulin test. So the leaf on the left here was the control. So these spores were not mixed with any RNA, and you can see the lesions developed quite nicely. This was actually 2 weeks after inoculation that the photos were taken. When the beta tubulin double-stranded RNA was added to the spores, you can see there's a few little pustules here, but really didn't see much disease at all on the leaves, which was really exciting. And we just wanted to test that it actually was the sequence specificity, the sequence-specific RNA that was having the effect. So we also tried treating some of the spores with GFP double-stranded RNA. So this is just the green fluorescent protein RNA, and the lesions developed similarly to the control. So it really looks like it was this rust-specific double-stranded RNA that was having the effect. And these are the full results. So actually it was quite promising. Most of the double-stranded RNAs that we tested had an effect at reducing the lesion development. These ones shown on the right were not significant, but the rest of these ones were. So it was just these few here that weren't, that didn't have a significant effect. So then we decided to move on to whole plant assays to see if it would also work on intact plants. So it was a similar setup, but this time Beck sprayed the double-stranded RNA and the spore solution onto the leaves rather than dropping them. And again, she assessed disease development 2 weeks after inoculation. So once again, the plant sprayed just with the GFP double-stranded RNA and without any RNA develops these pustules 2 weeks after inoculation, whereas these plants here, which were sprayed with the beta tubulin double-stranded RNA and the TEF double-stranded RNA, looked quite healthy. So Beck measured the disease proportion of the leaves, and you can see the proportion of disease leaves with the RNA treatment is really, really low. I think even beta tubulin, she hardly really even saw any pustule development. She also measured mean lesion size, but there wasn't much of a difference. Just going to the next one. So then she repeated this plant protection assay, but this time she actually sprayed the double-stranded RNA onto the plant 6 days before she inoculated them, which is sort of getting to the limit of what you can do with naked double-stranded RNA. But very excitingly, once again, she saw protection. So this is the beta tubulin sprayed plant here compared to the control, which you can see is covered in pustules. And once again, she measured the disease proportion of the leaves, and there was a significant reduction. The beta tubulin worked really well. And she actually saw a reduction in lesion size, but we have to repeat this experiment to see whether the lesion size is actually reduced. And this is some of our very, very new work, like we've only just done this recently. We were actually provided with this leaf mimicking substrate by Grant Smith at Plant and Food New Zealand. And I'd also like to acknowledge Volker Nock and Rebecca Soff from the University of Canterbury. And so this is actually just like a sort of polymer, but it mimics a leaf. And because it's quite hard to work with Myrtle Rust, because you can't really grow it in vitro, as you saw in our germination assays. It usually just sort of germinates and then doesn't really form any infection structures. Whereas on this leaf mimicking substrate, we're really excited because we actually saw cytoplasmic streaming occur. We saw aprosoria start to form, and we even saw infection pegs. So of course we couldn't help ourselves. We really had to try adding the double-stranded RNA to see if it would have an effect. And yes, we did see an effect. So without the double-stranded RNA, all of these infection structures started to form. This was 24 hours post-inoculation onto the substrate. But with the Rust-specific double-stranded RNA added, these infection structures were not seen. Sorry, this is the GFP control, which also formed the infection structures. And there was actually a reduction in germination, which we didn't see in our initial, well, we did see it, but it was quite variable in our initial in vitro tests. So we're going to do more experiments on this artificial substrate, but it really does seem to mimic a leaf surface because you really do get these infection structures. And it's quite promising for us to see that this double-stranded RNA, it really does seem to prevent the rust spores from forming those infection structures. So that's all I've got for today. We've still got a lot of work to do with this. So we have started doing some qPCRs to assess gene knockdown, and we're looking at it at different time points, and we need to know when to apply the double-stranded RNA for the best protection, so that qPCR will inform that. We also want to look into how long the double-stranded RNA persists in Austropuccinia psidii, as well as the Myrtaceae host plants, so Syzygium jambos, for example. But then we want to also move on to other Myrtaceae species which are actually at risk of extinction in Australia. So, for example, Decaspermin humili and Syzygium hopkinsoniae. And of course, we will test the bioclay to see if we can extend that protection window. So that's all very exciting work we're continuing. And I just wanted to end here with saying we are actually looking at some other pathogens, and I just wanted to mention we are also working on Phytophthora cinnamony, and we've had some promising results with that as well. So we've been doing some experiments on pineapples, and this is again in collaboration with researchers at the Queensland Department of Agriculture and Fisheries. So we've been growing the pineapples hydroponically. When we add the RNA targeting Phytophthora cinnamony into the water, we actually get some protection from the pathogens. So with the RNA, we get these healthy roots forming. Without the RNA, you get these brown lesions and the roots look really unhealthy. So we're following up with this work as well. So I'd just like to thank everyone at the University of Queensland, particularly Rebecca Degnan, the honours student who's done most of this work, Alistair McTarget, Chris Brosden, Bernie Carroll and Nina Mitter, Louise Shuey and Jeff Pegg from BAF, and Grant Smith, Volcanock and Rebecca Soft for the artificial leaf material. So that's all. Thank you, and I'll take any questions if there's time.
Thank you very much, Anne. That was excellent. And the question box is quite quiet, so do feel free to jump in there, people. But we do have one, and it is, are your RNA constructs modified in any way to promote stability?
So the naked RNA ones that I've been using aren't. So if we want to enhance stability, we will actually bind it to the clay and apply it to the plants as bioclay, but we haven't had to do that so far. We've only been doing lab assays, I should say. We haven't done anything out in the field yet.
Very good. And then the next question, and we are, of course, having lots of thank you for the presentation and excellent talk. Could you let us know, are there any human health risks and hazards to the operator, the person who's applying this, and the bystanders? Is there any toxicity expected to humans or aquatic vertebrates and vertebrates, as it does easily wash from the surface of leaves?
So I guess there is still some work going on looking into this, but so far it has been deemed safe to eat. We eat RNA all the time. As to it being sprayed onto you, I guess if you're the farmer in the field applying it, you would probably still wear some protective equipment because you wouldn't want to get sprayed with RNA. I don't think there's been much studies done on it, but you'd probably wear the same sort of gear you'd wear to spray on fungicides, and I don't think it would be as dangerous as spraying fungicides onto plants. Because we do have the barriers of the skin, obviously, and I mean, that protects us from RNA and other things in the environment. So, and for vertebrates, I think they have done some studies in mice and they've shown there's no effect, no harmful effect. But I think that is like an active field because people are getting a bit worried about if, you know, bucket loads of RNAs being sprayed around in the environment. But like for the actual target sequence, like I said, it's very specific. So we'll actually only target the pest or pathogen of interest. It won't actually harm any other organisms in the environment. It might just be if a large amount of double-stranded RNA caused some allergic response or something. I'm not sure. Yeah.
I see. Now, have you had any discussion on cultural acceptability of this technique in Australia? The person also says New Zealand. I doubt anything's happened in New Zealand yet. What about Australia?
Well, I was at the Myrtle Rust symposium in March, and we did have some discussions on RNAi vaccines with some of the Indigenous communities who were present, and some of them were actually quite excited about the idea that this could possibly be used to protect some of those very sacred trees that they have that were being affected by Myrtle Rust. But we would have to have a dialogue with them to see what the rest of the community felt, because that was only one specific group we were talking with about that.
Right, back to some more technical questions now. Can you give an indication of the average length and base pair of your double-stranded RNA?
Yep, so they're about 300 base pairs long, the ones we're using.
Okay, very good. Can you comment on how this enters the cell? Is it passive or active uptake?
Yeah, so that's still quite debatable, I think. There was a recent publication on sclerotinia showing that double-stranded RNA was taken up. It was endocytosis mediated, so it was sort of an active uptake into fungi. Into plants, No one's really sure. We think it might get into the leaves through stomata, but there's no evidence so far of it getting into cells or how it gets into cells. So we're not sure. There's still a lot of work going on into plants and where it goes in the plants. Is it in the apoplast? Does it move symplastically? Where does it get processed? Does it get processed? So yeah, that's still under debate. But for fungi, it seems to be endocytosis mediated.
Very good. Is there a risk for impact to other non-target fungi like mycorrhizal or endophytes? And I guess we've got a similar question.
We haven't tested that.
With beta tubulin is quite common. Have you tried to apply to other pathogens that are off target?
Okay, so yeah, no, we haven't, but that's where the bioinformatics comes into it. So the constructs we have been using were just designed against Myrtle Rust, but we actually have a sort of software pipeline here in the Myrtle lab where it's got all of the genomes of those beneficial organisms in there, and so any construct is sort of tested, aligned against those to make sure there's no matches. And so we can really design our constructs to make sure they won't target those organisms. But of course, we do need the genomes to be sequenced. So if there are organisms out there without the genomes available, it's possible we might accidentally target them. So I guess we would have to do some kind of study on the sort of communities on the leaves that we're going to be spraying or onto, or in the soil or whatever. I don't think it would be so much of a problem for the soil. In this case, we're targeting a leaf pathogen.
Mm-hmm. Sure, and just acknowledging we're going a few minutes over here, and now the questions are flowing in, but I think we'll keep going for another 5 minutes or so if that's okay with you, Anne.
Yep.
Right. Have you tested it on rust-infected plants as a treatment? So I guess they're saying rather than a preventative, has anyone tried spraying an already diseased plant?
No, we haven't tried that yet, but that's next on our list. That was one of the next experiments we're going to do.
Sounds like a good idea to me. If you looked at the influence of double-stranded RNA concentration on effectiveness of repression, and if so, can the double-stranded RNA production rates translate to field application requirements? Perhaps the question is, can we make this stuff as a commercial project?
Right, yeah. So Nina has actually been in discussion with some companies that synthesize or produce double-stranded RNA. I think they actually grow it in E. coli. They have big fermenters. And she's been told that they can produce very, very large quantities at very low costs. So commercially speaking, it will be quite doable, apparently.
So I guess that answers this latest question. The amount of RNA required, will this be economically transferable to agriculture or horticulture? I take it this product, well, I mean, obviously not this one, but this technique is not yet available commercially anywhere in the world, or are there actually products for sale?
Not that I'm aware of. So we have New Farm on board as our commercial partner, and they are currently looking into developing it in Australia. But as far as I'm aware, the topical application is not available anywhere. There are some GM transformed plants that function by RNA interference, but as far as I'm aware, there's nothing being applied commercially at the moment.
Very good. How long would the protection endure in long-lived trees?
Yeah, we're not sure. We also want to do some experiments just looking at RNA longevity in trees. So obviously we've been using seedlings, small plants at the moment, but we would ideally love to go and inject some double-stranded RNA into a big tree. We were actually discussing this more for avocado, more for horticultural tree crops. But yeah, we would love to check how long it persists in a tree. So that's something we will hopefully be able to do at some point, but we haven't done it yet.
So how long then is it lasting for the sprayed-on seedlings in the trials? I think you
So
said
we've
5
only
or so days.
tested, yeah, we've only tested 6 days. In some of our other tests we've been doing on other vegetable crops, we have seen double-stranded RNA still in the plant, like around 10 days. So I think once it gets in, it's quite stable. On the surface, it's not so stable, but if it's in the plant, it does seem to hang around for a while. So that's quite interesting.
Okay, what about toxicity issues with the bioclay, the application medium? Anything known
So
about that?
I think they're developing it so that there aren't going to be any toxicity issues. It is a natural sort of product, and it does degrade, but people I think are looking into, you know, whether anything builds up in the soil over time once it's degraded. But we're trying to make it as environmentally friendly as possible.
We're getting lots of potential issues raised here. Can the pathogen evolve some resistance to this? What happens if the target gene coding sequence gets mutated in the pathogen? Well, I guess then you have a problem, don't you?
Yeah, I mean, we haven't really had to deal with that yet, I guess. But like I said, we would be able to redesign our sequences. I'm not sure if
But like a vaccine, isn't it? You just make a new
I'm
one.
not sure if applying this double-stranded RNA would actually lead to the pathogen developing resistance, because that's sort of a different mechanism, you know. This RNA interference is really targeting the RNA. It's not like traditional pesticides which target different proteins.
It's not what I meant. Sorry to interrupt there. All right, we might perhaps have to have a follow-up by email
Okay.
with you about that, because yeah, we are really running quite over now. So yes, thanks. We'll get in touch about that. All right, I'm just going to ask 2 more questions. Are the target genes you're using uniform across the different strains of Myrtle Rust, or have you just worked on the pandemic strain?
Yeah, we've only worked on one so far. Yep.
Okay. Does the presence of the bioclay impact, for example, photosynthesis, because you must be having a layer on the surface of the leaf?
You can't really see much on the leaf, but I don't know if anyone's looked into that. It's possible,
Probably
but
not.
I think there'd still be enough sunlight getting into the leaves, and it's, you know, it wouldn't be like the whole plant was totally saturated with it. When you look at the leaves, you can only see a very maybe thin film on it. Like, I think light would still be able to penetrate really well. So I don't think so.
Well, the plants looked very happy to me. How long
Our plants always look quite happy. They don't look light deprived at all.
All right. Well, I'll give you one last comment from a person who's been an active participant in the question session. Great talk. Thank you. Exciting research. Smiley face. I think that all sums it up very, very well indeed. So that's it for today's talk. And yes, the video will be made available to everyone who registered. You'll receive that in your inbox in a couple of days. I also always have the videos on the Beyond Myrtle Rust website. Now, the next webinar will be Wednesday, June 9th at the same time, when we will hear from Mason Kamalani-Chock. He is a PhD student at the University of California in Berkeley at this time, but previously he was in Hawaii, where he was also working on a potential myrtle rust management method. We think you'd like to hear about that one too. And his talk is titled Harnessing Foliar Fungal Communities for Endangered Plant Conservation. Thanks very much, everyone, and we'll see you then.